Hi everyone, this will be our third practical and it's the aspirin assay that we'll be doing this week. So for this, our objectives are that you should be able to prepare a stock solution of acetyl salicylic acid, to then make a series of dilutions from your stock solution, and to use these dilutions to construct a standard curve for aspirin. You'll be given an aspirin tablet, which will be your unknown, and you should make a solution of that aspirin tablet and then use that to extrapolate the use your standards to extrapolate the concentration of aspirin in the aspirin tablet from the standard curve. So just by means of a general introduction, aspirin, or acetyl salicylic acid, is a weak organic acid that inhibits cyclooxygenase or COX enzymes in the body. Aspirin is normally rapidly deacetylated by esterases in the body to produce salicylate, which has the anti-inflammatory and also some antipyretic and analgesic effects. So the anti-inflammatory means that it reduces inflammation, antipyretic means it reduces temperature, and analgesic means it reduces pain. So the acetyl salicylic acid is hydrolyzed rapidly and quantitatively in basic solution to produce the salicylate diion and anion, and we'll see that in the next slide. So in the lab, this is what we do basically. So it gets rapidly hydrolyzed in a basic solution, which we use the sodium hydroxide for. This produces the salicylate dianion, and the mixture then is acidified by mixing with the solution containing iron 3 ions. This will then form an intensely purple complex, so it's a different version of the iron 3 ion, and this is what gives it its color. So normally aspirin solution is colorless. The reason why we mix it with the sodium hydroxide and then with the iron solution is to hydrolyze it to produce salicylate, and then to give it a purple color, so that we can quantify this then using a spectrophotometer. And remember, the intensity of the color is directly related to the concentration or the amount of aspirin present in that solution. So knowing that, we can make a stock solution of the aspirin and generate a series of standards of known concentration, which can then be quantified using the spectrophotometer and allow it to generate a standard curve. So this standard curve can be prepared using different known concentrations of the aspirin, and then we can use it to quantify or extrapolate the concentration of an unknown amount of aspirin from the curve. So this is essentially what happens chemically. We have the aspirin, and a complex then is formed by reacting the aspirin with sodium hydroxide. So the hydroxide ions then have the effect of hydrolyzing it. This mixture then that results is reacted with iron 3. So the iron 3 solution that we use reacts with the salicylate in solution, and it generates an intense purple color. And this intense purple color then is what we use to measure in the spectrophotometer. So from a practical point of view then, making the stock solution, you will have 10 mils of 1 molar NaOH in your box. You should put 10 mils of this into your square base containers, weigh out 200 milligrams of acetyl salicylic acid and add it to the NaOH. This is your standard solution. In addition, you'll also have your unknown, and your unknown this time is a tablet. So break up the tablet in a sheet of tin foil, make sure that it's well crushed, add it to the second container of sodium hydroxide. So this should be labeled sample. So 2 containers, one for a standard, which you'll make your standard series of standards from, and then the second one is your sample. Cap the containers, and then you'll have to vortex these for 10 minutes and leave them to stand for 30 to 40 minutes with occasional shaking to form the salicylate dianion. You should also check that all the powder is in solution and refer to your demonstrators or the academic present to check for this. Then transfer solution A, the acetyl salicylic acid, to 100 mils of to 100 ml volumetric flask, and this is a solution A, and then also do the same for solution B, which is your tablet. So in terms of the standards then, similar to the tartrazine practical, You'll prepare the standards in Eppendorf tubes from your stock solution A and fill in the values that you get into the table in your lab book. Get your demonstrator or academic to check while waiting for the aspirin to dissolve. For your sample then, you should have a neat, a 1 in 2 and a 1 in 5 dilution of your sample. So quite similar to the tartrazine practical where you had the neat and 2 dilutions then as well. So you'll need to label another set of Eppendorfs in addition to the ones that you had for the from above where you were generating the stock solution. This is because you're transferring it into new solutions. You will need 11 tubes, 1 for each standard and 1 for each of the samples. So neat 1 in 2 and 1 in 5 dilutions. And you pipette 50 microliters of each of the standards and the neat and the diluted samples into each of these Eppendorf tubes. On top of that then is when you add the iron solution. So 950 microliters of the 0.02 molar of the iron solution to each tube. Vortex all the tubes, make sure it's well mixed, and then once that's done, you pipette 200 microliters in quadruplicate, so 4 times for each standard and also for each of the samples, so your neat 1 and 2 and 1 and 5 into the 96 well plate. Finally then, you'll read your absorbance at 540 nanometers on the plate reader. Get your readout and to remember to subtract the blank from all of your other values, the average of the blank from the other values first. From that you can construct a standard curve similar to the tartrazine with absorption on the y-axis and concentration of the standards on the x, and construct a standard curve and extrapolate the concentration then of the aspirin in the tablet from the neat, which is your undiluted, your 1 and 2 and your 1 and 5. And don't forget to multiply the 1 and 2 concentration by 2 and the 1 and 5 concentration by 5. You then get an average concentration from these.